SwayamPrabha. Digital India. Educated India.
Welcome to the Proteomics course. In this module, we are discussing about mass spectrometry techniques. In the previous lectures, we talked about basic concepts of mass spectrometry. Then we discussed about Mod-22 MS, and now today we will talk about liquid chromatography mass spectrometry or LC-MS. MS is based on production of ions which are subsequently separated according to their M/Z ratio. The resulting mass spectrum provides a plot of relative abundance of generated ions as a function of M/Z. MS provides most versatile platform and comprehensive analytical technique for the proteomics scientist for a wide variety of applications. So, in today's lecture, I will first give you an overview of the mass spectrometry workflow. We will then talk about individual components in some more detail, such as liquid chromatography, ionization sources, mass analyzer, and then tandem mass spectrometry. So, as you can see in the slide, in the MS experiment workflow, there are multiple steps are involved. I have numbered those from 1 to 5. So, protein samples are digested using trypsin and peptide mixtures are fractionated by using liquid chromatography or LC. These fractions are subjected to an electrical potential which results into spray formation. In ESI or electrospray ionization, it leads to desolvation and ionization of peptides. The mass to charge ratio are measured in mass analyzer, which is shown in step 4. Specific ions are randomly selected in the collision cell, and then based on the collision induced dissociation, the resulting fragments ion are further measured in the second mass analyzer. The MS precursor ion intensity can be used for peptide quantification, and MS ion fragmentation can be used for the sequence information and protein identification. So, in this workflow, all these steps are equally important, starting from the first step, the tryptic digestion, second, pre-fractionation strategies using liquid chromatography, then ionization sources, mass analyzer, and then spectrum generation MS or MS. So, first step, in gel digestion. It is a multi-step procedure which includes a spot selection if you have taken the gels from the 2D gel, or if you have used the gel free techniques, you can still run the samples on the gel, excise the band, and then excise those bands. After that, you have to remove the stains if you had used with the Kumasi staining, then reduce, alkylate, and then perform the proteolytic cleavage, and finally, peptides can be extracted. This slide gives you an overview of various steps involved in the in-gel digestion. I have discussed this in more detail in the previous lecture while discussing about the Mod-22 MS. So, you can refer to more detail over there, but just to refresh you the concept, I will talk this concept again in the following animation of in-gel digestion. Electrophoretic separation of a protein mixture results in distinct protein bands. These proteins can be used for analytical purposes by carrying out in-gel digestion. Entire gel is fragmented into small pieces and each piece is dissolved in a suitable buffer. To simplify the complex proteome, it is good idea to chop down the overall separate proteins into the smaller pieces. The protein solution is treated with a reducing agent like dithioethritol DTT, which cleaves the disulfide bond in the protein. This is followed by treatment with iodoostamide or IAA, which alkylates the sulfhydryl group and thereby prevents the reformation of disulfide bonds. After cleavage of the disulfide bonds, the protein is treated with a proteolytic enzyme. The most commonly used enzymes include trypsin. Trypsin cleaves the protein at specific residues, arginine and lysine, and generates smaller peptide fragments. This tryptic digest is used for further purification and analysis. So, after doing the in-gel digestion, now let us move on to second part, the separation technology, liquid chromatography or LC. So, chromatography in general is a physical separation method in which components for separation are selectively distributed in two immiscible phases, a mobile phase flowing through the stationary phase. Now, depending upon the mobile phase, the technique is termed as either liquid chromatography or gas chromatography, etc. So, what is liquid chromatography? The peptide mixtures can be fractionated in line with the instrument prior to the introduction into mass spectrometer. That is, I think, one of the advantage for doing the proteomic applications. So, in LC, it can separate mixtures or components on the basis of differences in the affinity for stationary and mobile phase. Liquid chromatography is also useful for removing the undesirable impurities. It also increases reproducibility in the samples as well as robustness of the MS measurement. LC along with further enrichment can help into the concentration of the diluted samples. It also helps in increased sensitivity, detection of low level proteins, and further it can separate peptide mixtures. So, there are various types of chromatography one can use for different applications. In proteomics, the most commonly used method for the peptide fractionation is reverse phased liquid chromatography or RPLC, which separates peptides based upon the hydrophobic binding of interaction between the peptides or proteins in the mobile phase and immobilized hydrophobic ligands in the stationary phase. By utilizing this hydrophobicity, one can separate the peptides. However, if your proteome mixture or the peptides are very complex, then one can further use another type of chromatography method such as strong cation exchange as well as different type of multi-dimension separation can be employed. In this slide, I have shown you the configuration for reverse phase HPLC. As you can see, there are two components in the mobile phase A and B linked with the HPLC pump. A buffer can provide between the range of 0.1% of formic acid to 5% of acetonitrile, and in B 0.1% to 80% of acetonitrile. So, first of all, equilibrate the system in buffer A, then load the peptides and wash those. Then run the gradient of increasing mobile phase B. Now wash the reverse phase with buffer B and then re-equilibrate in A. In this way, the peptides can be separated, and prior to MS analysis, it can be desalted so that there is no interference of salt. Now, reverse phase is most commonly employed with the electrospray ionization because of its compatibility of reverse phased acidic aqueous and polar mobile with electrospray ionization. As I mentioned, in the proteomics, one can use ESI and liquid chromatography in line. So, directly samples can be pre-fractionated and further analyzed using mass spec. The inline reverse phase HPLC is very useful because it can do the desalting of peptides prior to ionization in ESI. There is no need for doing separate offline desalting and pre-fractionation. It can focus peptides from the dilute samples into the narrow chromatographic bands, and it also enhances the sensitivity. Let us now talk about another separation technique which is also commonly employed in proteomics, which is the strong cation exchange or SCX. So, in SCX resin, the silica based cation exchange used in the stationary phase. The sulfonic acid cation based exchange ligands are used. These ligands are covalently bound to the polymeric coated silica. Now, as I have shown in the slide, there are two important phenomena here. One is retention, other is elution. In retention, that is based on the electrostatic attraction between the negatively charged sulfonic acid and positively charged peptides. The elution can be performed by an exchange of peptides for cation of mobile phase additive, the ammonium ions, and this is a reaction to the high concentration of cations. Now, HPLC can be microcapillary, it can be nano LC, or it can also be different type of chip based chromatography separation. Now, for proteomics, various type of microcapillary, nano LC and different type of chip technologies are currently used. In the microcapillary HPLC, the low flow rate, which is less than 1 microliter per minute, is more sensitive as compared to the standard reverse phased HPLC, which is around 50 microliters per minute. The microcapillary HPLC is required for the analysis of low femtomole amount of the peptides. One can prepare the microcapillary HPLCs by using few silica capillaries and then pack that with the reverse phased packing material. For pre-fractionation, multi-dimension separations are used. The different type of principles are involved for separating these peptides. One can use size exclusion chromatography, which separates based on the molecular weight or the size, ion exchange chromatography based on the charge, capillary electrophoresis based on charge, reverse phased based on the hydrophobicity, affinity chromatography, which is based on the biological interactions. Now, multidimensional approaches can be coupled with the mass spectrometry. So, one can use various type of liquid chromatography methods in tandem and then do the multidimensional separation. Different type of approaches have been tried. For example, size exclusion chromatography followed by reverse phase chromatography, but it has resulted into the poor resolution of peptides in size exclusion chromatography. So, that is not very widely used. Other approaches include reverse phase chromatography followed by the capillary electrophoresis, or size exclusion chromatography followed by the capillary electrophoresis. Due to the limited loading capacity and low loading volume of the capillary electrophoresis, again it is not very popular. The affinity chromatography based separations such as IMAC or Avidin followed by the reverse phased chromatography, those are commonly used, and a strong cation exchange followed by the reverse phased is most commonly used. So, this slide gives you an overview of various type of methods liquid chromatography separation which can be employed prior to injection into the ionization source. As I mentioned, the strong cation exchange as well as reverse phase chromatography together can be employed for various proteomic application, which have been demonstrated in the multidimensional protein identification technology or MUD-PIT. In this technique, the strong cation exchange separates by the charge, which provides low resolution fractionation in the beginning, and then reverse phase C18 column, which separates peptide based on the hydrophobicity, and it provides high resolution gradient. Let me describe some of the concepts involved in the liquid chromatography in following animation. I will also discuss MUD-PIT and some of the chip based approaches which are integrated for proteomic application with the liquid chromatography. So, let us discuss these concepts in following animation. A typical liquid chromatography setup consists of solvent bottles, degasifier, dual or quaternary pumps, sample injector, column and detectors. Here you can see the various components which are involved in performing the liquid chromatography. Different solvents can be placed in the solvent bottles depending upon the purification requirements. These solvents are mixed in the desired ratio and pumped into the column during elution after removal of any trapped air inside by means of the degasifier. The sample injector system may be automatic or manual. The automatic sample uses a syringe to inject the sample, which is placed in a vial directly into the column. Once the sample is injected, mobile phase flows into the column through the pump. The column consists of a stationary matrix that preferentially binds certain analytes. The outlet from the column enters the flow cell where it can be detected. There are various stationary phase matrices are available that separate the components of the mixture based on different principles. One of the most commonly used matrices include the strong cation exchanger or SCX, which separates charged peptides based on their electrostatic interactions with negatively charged sulfonic acid groups on the resin surface. Now, elution can be caused by the addition of positively charged mobile phase. The reverse phase chromatography is another commonly used tool, which uses a hydrophobic matrix consisting of long aliphatic carbon chains. These resin analytes separate on the basis of hydrophobic interactions and can be eluted by changing the polarity of the solvent. The nanoliquid chromatography, which makes use of C18 capillary columns, has gained popularity for the proteomic studies due to their ability to achieve finer separations. Now, these separated components pass from the column outlet into the flow cell, which is present in the detector. The most commonly used detector for protein analysis is UV detector, which analyzes the protein absorbance at 280 nanometer and plots a graph of retention time against intensity. Multidimensional protein identification technology, or MUDPIT, is a widely adopted strategy that carries out two consecutive protein separations based on different principles, as shown in the animation here. Shown on either side is a protein with different properties. Earlier we have talked about how one can make use of different properties of strong cation exchange and reverse phase chromatography to separate out peptides. Now, use those basic concepts and drag and drop the proteins that will interact with the SCX and RP regions of the columns respectively. So, your answer is correct. You can use the protein properties and separate those in the multidimensional protein identification technology. MUDPIT is a non-gel technique to separate and identify individual components of complex proteins and peptide mixtures of a proteome. It has been shown that MUDPIT has potential to be used as a substitute of traditional two-dimensional gel electrophoresis, since it can separate peptides in the 2D liquid chromatography. MUDPIT technique allows greater separation of peptides, which can directly be interfaced with mass spectrometry ionization source. It also avoids band broadening, which is one of the drawbacks of many chromatographic methods. We have discussed different type of liquid chromatography systems. In a traditional nanoflow LC-MS system, there are several fittings and connections are required, which is one of the major limitations of using those traditional systems. There are certain commercial advancements available to improve these type of limitations. Agilent's HPLC chip technology is a microfluidic device which carries out nanoflow high performance liquid chromatography and reduces limitations of several fittings and connections. The microfluidic device contains circuits of tiny closed channels and wells which are etched onto a glass or plastic microchip. Different forces such as pressure or electrokinetic can push a small volume of fluids in a defined manner. This technology integrates functional components onto a reusable biocompatible chip, which integrates sample enrichment and analytical nanocolumns, nanospray emitter, fittings and connection capillaries on a reusable biocompatible polymer chip. The chip based technology reduces the possibility of leaks and dead volumes. It improves sensitivity and reliability during the liquid chromatography steps. By making the integrated system, it avoids several pitfalls which are associated with separate tubings and fittings. Another important component of this technology is the HPLC chip MS interface. A chip is inserted into the interface which mounts on a mass spectrometer. So, this design configuration ensures that the electrospray tip is in the optimal position for mass analysis when the chip is inserted in the mass spectrometer. So, compared to the conventional nanospray techniques, this technology can achieve maximum sensitivity with minimum sample size by integrating sample preparation, separation and electrospray tip on the single chip technology. So far we have talked about trypsinization or doing the peptide cleavage. Then we talked about pre-fractionation strategies, liquid chromatography. Now, these samples are ready to be injected into the ionization sources. In the previous lecture, when we talked about basic concepts and an overview of the different type of procedures involved, I gave you a list of various type of ionization techniques available. We also discussed that soft ionization techniques are required for the proteomic applications. So, soft ionization techniques such as electrospray ionization ESI and matrix assisted laser desorption ionization or MALDI, these were introduced in the late 80s and now they have overcome the problem of hard ionization and now these are widely adopted for the proteomic applications. So, we will focus more on to the ESI and MALDI. Since we discussed MALDI in more detail in the previous lecture, I will focus more on the ESI or electrospray ionization in today's lecture. So, ESI, it requires sample of interest to be in solution and that is why we mentioned that we can use the inline separation along with the liquid chromatography. To ionize the samples, high voltage is applied to high conductively coated needle. So, this voltage results into the sample becoming charged either positive or negative. The positive ions are primarily used for the analysis of proteins. The distinguishing features of electrospray ionization includes its ability to produce multiple charged ions. The number of charges that can be accepted by a particular molecule depends on its basicity and its size. Now, here you can get an overview of the process involved in the electrospray ionization. The small droplet of solutions are generated by the Taylor cone which contains the peptide analyte. Protons from the acidic solution provides droplets the positive charge, so that it can move from the needle to the negatively charged instrument. In ESI, the desolvation of ions occur at the atmospheric pressure and mass analyzer is maintained at a lower pressure. So, that ions can be drawn into the MS based on the pressure differential. During movement, the evaporation reduces the size of droplets and then it splits into the small charged droplets. Ions when enter inside the mass spectrometer, the droplets are dried using a vacuum of the inert gas, which results into a gas phase ion acceleration through analyzer towards the detector. You can see this process with much clarity here in this slide. The top panel is showing the Taylor cone generation. and the center it is shown that production of multiple charged ions. Usually it is coupled to the MS via real time liquid separation. After discussing three important components, now let us move on to the fourth part, the mass analyzers. There are different type of mass spectrometers currently available, but for proteomics there are two configurations which are most commonly or most oftenly used: the quadruple time of flight or QTOF based configurations and hybrid linear ion Orbitrap instruments. The TOF configurations separates peptides in time as they reach on the detector. So, the time of flight is measured, whereas the Orbitrap mass analyzers, they measure frequency of peptide ions which are oscillating in the ion trap. Now, different type of resolution and sensitivity can be obtained from each of these configurations. In the previous lecture, I gave you an overview of different type of mass analyzers currently available. Each of those have its own unique properties in mass range, analysis speed, resolution, sensitivity, the ion transmission and dynamic range. The time of flight analyzers use time flight, ion trap, Orbitrap and ion cyclotron resonance. They separate ions based on their mass to charge resonance frequency, whereas quadruples or Q, they use oscillating electrical field for selective stabilization of ions. This just gives you an overview of various type of mass analyzers, and briefly we discussed about their principle. Now, mass analyzers can be categorized broadly into the scanning MS, ion beam MS and trapping MS. Scanning MS is more commonly used with the TOF, which is further coupled with the mild deionization sources. The ion beam MS is commonly used for the quadruples, whereas trapping MS for the ion traps, Orbitraps and FTICR. All these can be coupled with the electrospray ionization ESI. Now, some of the important mass analyzers, let us discuss a little more detail. First, talk about time of flight, which is one of the simplest mass analyzer currently used in combination with the MALDI. The TOF has emerged as one of the mainstream technique for the analysis of biomolecules, and it is widely used for various applications. In TOF, the ions are accelerated to high kinetic energy, and due to their different velocities, they are separated in a flight tube. One can also use the reflectron mirror, so that ions can turn around into a reflector, and it can compensate for minor differences in the kinetic energy and provides long separation. Another commonly used mass analyzer is quadrupole. The Q instruments are one of the most widely used type of mass analyzers currently used in proteomics. It consists of 4 matched parallel metal rods, and mass separation is accomplished by the stable vibratory motion of ions in a high frequency oscillating electric field that is created by applying direct current and radio frequency potentials to these electrodes. So, as we talked, quadrupole is set of 4 parallel metallic rods with opposite pairs are electrically connected. The different modes one can use for this analysis: RF or radio frequency mode, which allows ions of any m by z ratio to pass through; scanning mode, ions of selected mass by charge can be allowed by the detector. The potential difference applied, an instrument can be used as a mass filter. the neutral loss scan and precursor ion scanning method. They are used for the phosphorylation to distinguish the phosphorylated and non-phosphorylated peptides. Now, triple quads, which is arrangement of quadruples, is widely used for the proteomics. In triple quad, the Q1 scans ion streams. It directs ions of a selected m by z ratio into the second quadrupole, Q2, which is a collision cell. So, as you can see in this slide, the collision cell operates in the radio frequency mode. The fragmentation of intact peptide ions can be induced by colliding with inert gases, and then selected ions are further moved into the Q3. Q3 scans the stream of ion fragments which are emerging from the collision cell to generate a collision induced dissociation spectrum. The mass spectrum of fragments derived from one peptide after one analysis is complete. Then Q1 directs a different intact peptide into the collision cell. So, in this sequential manner, it can process various peptides. Now, let us talk about another important mass analyzer, ion trap. The ion trap traps ion using electrical fields, and it measures by selectively ejecting them to a detector. It consists of a chamber which is surrounded by a ring electrode and two end cap electrodes, as you can see in this figure here. The voltage applied to the ring electrode determines which ions remain inside the trap. So, ions above a threshold of m by z ratio, they remains inside the trap, and others can be ejected through a small hole. Theoretically, ion trap can provide MSN analysis, and it can also provide a mass filter. One important mass analyzer is Fourier transform ion cyclotron resonance, or FTICR. Due to its high resolution and MS-MS capabilities, application of FTICR MS in combination with electrospray ionization has been employed for the large biomolecules, and now it is also used in the proteomics. An FTICR MS can be considered as an ion trap system, where ions are trapped in the magnetic field. It uses cyclotron motion or cyclotron frequency to resolve the ions. Although operationally it is very complex and not very easy to operate, but it provides highest resolution, mass accuracy and sensitivity. It also provides the capability of multiple tandem experiments, and MS of very large ions are possible by using FTICR MS. So, we have discussed all the important components of liquid chromatography mass spectrometry. Now, one can apply these configurations in tandem. One can select different type of mass analyzers and use it based on their applications. So, now we will look at some of the popular hybrid MS and MS-MS configurations. Multi-TOF, that is one of the widely used tandem MS configuration. In this one, the TOF or two time of flight tubes as well as hybrid quadrupole time of flight analyzers can be used. We have discussed the multi-TOF system in some more detail in the previous lecture. So, I will move on to some other configurations, which is QTOF. The QTOF, it combines front part with quadrupole, or it can be triple quad TQ, along with the TOF analyzers to measure the mass of ion. Some of the important concepts involved in ionization, mass analyzers and tandem MS. I will describe those in following animation. Tandem mass spectrometry. Let us first discuss the individual components, the ionization source, mass analyzers, and then how one can make use of various type of ionization sources and mass analyzers in tandem for the mass spectrometry analysis. The ionization source is responsible for converting analyte molecules into gas phase ions in vacuum. This has been made possible by the development of soft ionization techniques like matrix assisted laser desorption ionization (MALDI) and electrospray ionization (ESI). In MALDI, the analyte of interest is mixed with an aromatic matrix and bombarded with short pulses of laser. The laser energy is transferred to the analyte molecules, which undergo rapid sublimation into gas phase ions. Another electrospray ionization technique, ESI. In ESI, the sample is present in the liquid form and ions are created by spraying a dilute solution of the analyte at atmospheric pressure from the tip of a fine metal capillary, which creates a mist of droplets. These ions are then accelerated towards the mass analyzer depending upon their mass to charge ratio. Now, let us discuss about mass analyzers. The mass analyzer resolves the ion produced by the ionization source on the basis of their mass to charge ratios. There are various characteristics such as resolving power, accuracy, mass range and speed, which determine the efficiency of these mass analyzers. The commonly used mass analyzers include time of flight, TOF, ion traps, quadrupole, ion cyclotron resonance, orbitrap, magnetic sector, etc. The time of flight analyzer accelerates charged ions generated by the ionization source along a long tube known as the flight tube. Ions are accelerated at different velocities depending on their mass to charge ratios. Ions of lower masses are accelerated to higher velocities and reach to the detector first. The TOF analyzer is most commonly used with the MALDI ionization source, since MALDI tends to produce singly charged peptide ions. The time of flight under such circumstances is inversely proportional to the square root of molecular mass of the ion. An ion trap makes use of a combination of electric and magnetic fields and captures ion in a region of a vacuum system or tube. It traps ions using electric fields and measures the mass by selectively ejecting them to a detector. Quadrupole mass analyzers. They use oscillating electrical fields to selectively stabilize or destabilize the paths of ions through a radio frequency or RF quadrupole field. The RF mode allows ions of any m by z ratio to pass through. So, there are two modes in which the quadrupoles can be operated, the radio frequency or the scanning mode. The RF mode can allow all the ions to pass through, whereas the scanning mode, the quadrupole analyzer selects ions of a specific m by z value as defined by the user. A range can also be entered, in which case only specific ion satisfying the criteria moves towards the detector and rest can be filtered out. The triple quadrupole consists of two sets of parallel metallic rods interspersed by a collision cell. The first quadrupole scans the ions coming from the ionization source and allows only ions of a particular m by z ratio to pass through. Once the ions are selected, these ions enter the collision cell, where they are fragmented by collision against an inert gas like argon. The smaller fragments then enter the third quadrupole, which scans all the ions in a radio frequency or RF mode to generate a spectrum based on the varying behavior of ions in an oscillating electrical field. The different type of tandem MS-MS configurations, such as Multi-Toff-Toff. The Multi-Toff-Toff MS is a common tandem MS configuration, in which the ions are first resolved on the basis of their time of flight in the first Toff analyzer. The selected ions enter the collision cells, where they are further fragmented. The fragmented ions are accelerated and further resolved on the basis of their m by z values in the second time of flight tube, after which they can be detected. ESI-QTOF is another common tandem MS configuration that first selects ions in radio frequency mode. Selected peptides are fragmented in collision cells and resulting ions are accelerated and resolved on the basis of their time of flight. So, finally, there are so many mass spectrometers currently available commercially. So, now depending on individual's application, one can select different type of configuration. Based on an excellent review from Yates and colleagues, I have provided this performance comparison of MS instruments in following slide. Here you can see the linear ion traps or LIT or LTQs. They have resolution of 2000, mass accuracy 1000 ppm, mass accuracy 100 ppm, sensitivity femtomole and scan rate is very fast. The triple quadrupoles or TQs with resolution of 2000, mass accuracy 100 ppm, sensitivity in attomole and scan rate is moderate. The LTQ or B traps, they can provide high resolution 100,000, mass accuracy 2 ppm, sensitivity in femtomole and scan rate is moderate to low. LTQ, FTICR, they can provide very high resolution of 500,000, mass accuracy less than 2 ppm, sensitivity in femtomolar range, slow scan rate. The quadrupole time of flight, they provide resolution more than 10,000, mass accuracy 2 to 5 ppm, sensitivity in attomole range, and scan rate is moderate to fast. I hope in today's lecture you got various concepts together how one can use in mass spectrometry experiment. We have discussed individual components of in-gel digestion, liquid chromatography, ionization source, mass analyzers, and how they can be used in tandem. And then finally, we compared various commercial configurations currently available, so that one can get an idea that what type of resolution, sensitivity, scan speed, etc. they can provide. We will continue our discussion on mass spectrometry and data analysis in following lecture. Thank you.